Good morning, children. Have a nice and wonderful day. Let us start today the topic nucleic acids. This topic comes under the molecular basis of inheritance of standard 12. This topic is very important for board as well as NEET examination. So be attentive in the class and concentrate. Have a look towards the board, my dear children. The topic name is written, that is nucleic acid part 1. I'll be discussing nucleic acid, the topic nucleic acid, in two phases. In first page, I'll be discussing nucleic acid part 1, I have written here. I'll be discussing the general concept, the various terms that are being used in nucleic acid, and we'll be recapitulating the topic what we have studied in standard 11 Biomolecule chapter. Okay, I'll be discussing DNA in part 2 in detail, that is the structure of the DNA and the polynucleotide chain which is there in molecular basis of inheritance of standard. Nucleic acid part 1 I am going to start, my dear children. Now listen to me carefully and welcome to my classes. Nucleic acid, the name nucleic acid is given because it is mainly found in the nucleus and acidic in nature. The credit of discovery of nucleic acid goes to F. Mischer, a Swiss biochemist and physician. He discovered the nucleic acid, mainly the DNA, in 1869 and named it as nuclein. Very important for examination point of view. Now children, next point to see the types of nucleic acid. Through chemical analysis of chromosome, it was found that there are two types of nucleic acid, that is DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Both the nucleic acids are found in all living system or cells, and we cannot think of life without these two nucleic acids. Clear? So these two nucleic acids are essential for survival of an organism. Now recall viruses, my dear children, one exception is there, where viruses having only either DNA or RNA, never both. That's why viruses form a connecting link between living and non-living. Clear? Now next point is nature of the nucleic acid. Both nucleic acid and DNA and RNA are acidic in nature, negatively charged, large molecule, means they consist of many monomeric units called as nucleotide. You know that is nucleotide is the unit of nucleic acid. And apart from that, both nucleic acid show your optical activity, my dear children. Okay. Now come to the site of the nucleic acid. Nucleic acids mainly found in the nucleus. Clear? Apart from that or besides that, nucleic acids are also found in mitochondria, chloroplast, and even in the cytoplasm of the cell. But the main site or location of nucleic acid is nucleus. Next to your function, DNA and RNA, these are two types of nucleic acid. DNA is mainly the genetic material in almost all living, but RNA is genetic, but RNA is genetic as well as non-genetic, my dear children. But largely or mainly, RNA is non-genetic and helps in protein synthesis. But only in the absence of DNA, RNA directly acts as genetic material, as in some RNA viruses, that is riboviruses. Examples are most plant viruses like TMV, a few animal viruses like influenza viruses, and bacteriophage, QB bacteriophage. So QB bacteriophage having RNA, TMV also having RNA. Please remember these two examples at least. QB bacteriophage is very important because all bacteriophages having your DNA you have studied in standard 11. But this is the only bacteriophage, QB bacteriophage, having your RNA, my dear children, okay? Non-genetic. So non-genetic RNAs, they mainly help in protein synthesis. They are of three types. I have written it. mRNA, tRNA, and your rRNA. Now among this mRNA means your messenger RNA, tRNA call your transfer RNA, or it is also called your soluble RNA or sRNA, and then the last one is rRNA, that is your ribosomal RNA. Clear? All these three RNAs, they play vital role in protein synthesis. Now one important point you should remember, messenger RNA. Messenger RNA is the most important RNA. If question comes among these three RNAs, that is mRNA, tRNA, rRNA, which one is the most important RNA? That is mRNA would be the answer. Okay. Next, tRNA is also called adapter molecule, transfer RNA. Why? Why it is called adapter? It picks up amino acid from the cytoplasm and brings to the ribosome for protein synthesis. Clear? RNA, that is ribosomal RNA. There are structural RNA. They also help in rRNA, also helps in protein synthesis. They mainly form the structure of the ribosome. Ribosome is formed by your rRNA. rRNA plus protein forms your ribosome. Clear? So that's why rRNA is also called structural RNA. Getting me? You understand? Now, rRNA actually formed from the nucleolus of the cell. Nucleolus of the cell. Question, very important question in rRNA, that is rRNA is formed in the nucleolus of the cell. Clear? That's why nucleolus sometimes called it, or it is generally called it the ribosome factory. Clear? So ribosome factory, answer will be nucleolus, but protein factory is called your ribosome. We have studied, my dear children, in cell in standard 11. Clear? Very two important questions I have told you. Now, rRNA also having your catalytic function, enzymatic function. They behave like enzyme. Clear or not? Call your ribozyme or RNA enzyme. One of the example of this RNA is 23S rRNA in bacteria. It is 23S rRNA in bacteria or prokaryotes and 28S rRNA in eukaryote. These two RNAs, they help in peptide bond formation. Now, one of the example of this ribozyme we will be studying in upcoming chapter of protein synthesis, that is peptidyl transferase. Peptidyl transferase is an example of RNA enzyme or ribozyme. Clear? This enzyme, peptidyl transferase, helps in peptide bond formation during protein synthesis. Now, this RNA and DNA, my dear children, they work in a coordinated manner, in an integrated manner, in a regulated manner. See the chart I have given below, DNA to RNA, RNA to proteins, proteins to enzyme. Clear? And enzymes, they control the metabolism, that is catabolism and anabolism, and finally they help in expression of a trait. So how regulated, how they are working in a regulated manner, the DNA and RNA. You can understand. This DNA to RNA is called transcription, RNA to protein, that is called translation. Transcription is also called coding, and translation is also called decoding. These two terms will come across in our upcoming chapters of protein synthesis. Now children, let us discuss the chemical composition of nucleic acid, that is both DNA and RNA. Both DNA and RNA are chemically composed of, see the board, chemically composed of nitrogenous bases, sugar molecules, and phosphoric acid molecules. Now come to the first point, that is nitrogenous bases. There are two groups of nitrogenous bases, purines and pyrimidines. Purine bases are adenine, guanine, and pyrimidine bases are cytosine, thymine, and uracil. Okay, now purine bases have double ring structure. Purine bases have double ring structure, see the figure, that is dicyclic, whereas pyrene bases having your single ring structure, that is monocyclic. Over. Now the next point. Next point is carbon and nitrogen having your 9-member ring. Carbon plus nitrogen, 9-member ring. You see 9-member ring is there, numbering 1, 2, 3, 4, 9. Here also carbon and nitrogen 6 member ring. Very, very important. The numbering I have done here. So here, from here 1 starts. 1, 2, 3, 4. Now in pyrimidine 1 starts from here. Many children do the mistake. So position you should know. Now simple general structure I have given about the purines and pyrimidines. Detailed individual structures of adenine, guanine, cytosine, thymine, uracil you have studied in biomolecule chapter in standard 11. So only for your convenience I have given the general structure so you can memorize. Now the next point will be nitrogen at 1379 position in purine bases. Very, very important, my dear children. Here in pyrene bases, nitrogen is found at 13 positions. Clear? Now, about the sugar molecule. Sugar molecule in both, that is your DNA and RNA. RNA has got ribose sugar, DNA has got deoxyribose sugar. Both the sugar molecules are pentose sugar, that is having 5 carbon. Both are aldose sugar. Here I have written pentose, aldose, that is pentose means 5 carbon, aldose means having aldehyde group, that is aldopentoses. Now in ribose and deoxyribose, see the differences in the structure. Both are pentose sugar, 5 carbon, 4 carbons in both cases are within the ring and the 5th carbon is outside the ring. Now children, here you see at 2 carbon of the ribose sugar, OH group is there. Means oxygen is there. Oxygen atom is there. That's why it is more reactive and less stable. Very, very important. Clear? Now in deoxyribose you see it is formed from the ribose sugar. At 2 position OH group is there. 1 atom of oxygen is removed from ribose, making it deoxyribose. So here you see there is no oxygen. When there is no oxygen, so deoxyribose sugar is less reactive and more stable. The question is asked, which nucleic acid is more stable? That is, DNA is more stable and then your RNA. DNA is more stable than RNA. Remember this thing. Now come to your phosphoric acid. Phosphoric acid or phosphate molecules are present in both the nucleic acid. Because of this presence of this phosphoric acid, the nature of both the nucleic acid is what? Acidic as well as negatively charged. I have already explained earlier in the beginning. Now see how the oxygen atom are negatively charged in phosphate. So it is negatively charged and acidic in nature. Now there are some important questions you can see. That is your the thiamine base which is present. Thiamine. Another chemical name of thiamine base is your 5-methyl uracil. Very, very important for NEET and board examination. If statement comes that another chemical name of thiamine is what? 5-methyl uracil. Okay. Now which one is more stable, RNA or DNA? DNA is more stable than RNA. Clear? Now in RNA, which group is there? I have written here, see, RNA possesses addition of dash group at dash position. Which group? At 2 carbon, that is your OH group is there. That's why when oxygen is present, it is more reactive and less stable. Very, very important. Now children, I have written one note here below. There is sugar and phosphate which are there in nucleic acid. They form the backbone of the nucleic acid. This is all about the chemical composition in brief. You have studied this same thing in standard 11 of the chapter Biomolecules. Okay? Now I will be discussing nucleoside, nucleotide and polynucleotide chain. Have a look towards the board. Nucleoside. Nucleoside means base, nitrogenous base plus sugar is called nucleoside. Clear? Now this is the structure of the sugar molecule. Here the base is attached at 1 carbon or to the 1 carbon of the sugar by a bond or linkage called your N-glycosidic linkage. Are you getting me? So base is attached to carbon number 1 of the sugar molecule with the help of a linkage or bond called N-glycosidic linkage. Clear? Now there are different type of nitrogenous bases. You see, that is adenine, guanine, cytosine, thymine, uracil. These are the bases. Now in DNA, the nitrogenous bases are adenine, guanine, cytosine and thymine. Whereas in RNA, the nitrogenous bases are adenine, guanine, cytosine and uracil. So the total number of nucleoside will be 8. 4 of your DNA and 4 of your RNA. Have a look towards this. If your adenine base is attached with sugar, then the nucleoside is called adenosine. Similarly, guanosine, cytidine, thymidine and uridine. These are the types of nucleosides. Clear? Are you getting me? Is it clear? Do you understand? Okay. So total number of nucleosides will be 8. 4 of DNA and 4 of RNA. Now this nitrogenous base is attached to carbon 1 of your sugar molecule with the help of N-glycosidic linkage. So there are 2 groups of nitrogenous bases, purines and pyrimidines. In purine base, there is adenine, guanine. They form 1 carbon with 9 position of nitrogen of purine bases. So 1,9, 1C9N glycosidic linkage in purine. Clear? In case of pyrimidine, the N-glycosidic bond is formed between 1 carbon and 1 nitrogen of pyrimidine base like your cytosine, thymine and uracil. Of pyrimidine. Very, very important, children. Now come to nucleotide. Nucleotide means base plus sugar and phosphate. They form the nucleotide. And nucleotide I have already discussed with the unit of nucleic acid, that is both DNA and RNA. Now this is the sugar molecule. I have explained that this is the N-glycosidic linkage formed between 1 position of the carbon with the nitrogenous bases. This is very important. Now after that, at 5 carbon, there is phosphate is attached and the bond is called phosphoester bond or phosphoester linkage. Clear? Now I have written that phosphate forms ester bond, that is a covalent bond with 5 carbon of the sugar to form a complete nucleotide. So the complete nucleotide is formed when base is attached to 1 carbon and phosphate is attached to the 5 carbon of the sugar molecule. Now types of nucleotides. Nucleotide types I am discussing. That is, nucleotides are mainly nucleoside plus phosphate. This is nucleoside, sugar plus base. And when phosphate is being attached, then that is nucleotide. So nucleoside plus your phosphate is called nucleotide. Now see the types of nucleotides. In DNA, the types of nucleotides AMP, GMP, CMP and TMP. Clear? D stands for deoxyribose sugar. Similarly, RNA, AMP, GMP, CMP, UMP will be there. So total nucleotides will be 8 nucleotides. The question is asked, how many nucleotides are there in a cell? The answer will be 8, my dear children. Now this AMP means that is nucleoside plus phosphate, adenosine plus phosphate, that is adenosine monophosphate. Guanosine is a nucleoside, guanosine plus phosphate, guanosine monophosphate. Clear or not? Then similarly cytidine monophosphate and thymidine monophosphate. We can call it adenylic acid. This is guanylic acid, GMP, cytidylic acid and thymidyllic acid. Or you can say AMP, GMP, CMP and TMP. D stands for deoxyribose sugar. Clear? So nucleotide means, children, tell me, that is nucleoside plus your phosphate. And the total number of nucleotides in a cell is 8 nucleotides. 4 of DNA and 4 of RNA. Clear now that I have explained? Now come to polynucleotide. Polynucleotide is a polymer or chain of nucleotides where many nucleotides they link or join to form a polynucleotide chain or a polymer of DNA and RNA. Clear? And the bond that joins two nucleotides is called phosphodiester bond or phosphodiester linkage. I have written. Where phosphodiester linkage means the bond that involves two ester bonds. Clear now? This is all about the nucleotide, nucleoside and polynucleotide. Clear, children? Now children, biomolecules. Biomolecules related to nucleic acid. Now have a look towards the board, my dear children. Adenine. Adenine is a base, nitrogenous base. Adenosine, that is base plus sugar, that is adenine plus sugar. Similarly, AMP, that is adenosine monophosphate, means base plus sugar plus phosphate. Now ADP, base plus sugar plus 2-phosphate, ADP, adenosine diphosphate. Now look towards the board. ATP, adenosine triphosphate, means base plus sugar plus phosphate plus phosphate plus phosphate, means 3-phosphate. Now FAD, base plus sugar plus phosphate whole bracket 2, that is base sugar and phosphate. FAD is flavin adenine dinucleotide. So it is a dinucleotide, my dear children. Clear now? Now one question comes from this biomolecule, what I have written here. Arrange the following biomolecules according to their increasing molecular weight or from simplest to largest in an ascending order or in a descending order. These are the questions. Is not it in NEET examination or board examination? Clear? Now important points are highlighted here. Have a look towards the board, my dear children. One is single nucleotide. Single nucleotide means one nucleotide may contain 1 to 3-phosphate. I have written. Clear or not? So one nucleotide may contain 1 to 3-phosphate. Clear? When more than one phosphate is present in a nucleotide, then they are called as nucleotides of higher order. Example, nucleotides of higher order, question may be asked. That is your ADP and ATP. Clear? In ADP there are 2-phosphate and in ATP there are 3-phosphate. So these are the examples of nucleotides of higher order. Clear? Now ATP, you know, it is an activated nucleotide, adenosine triphosphate, which is said to be the energy currency of the cell. You know this thing. And now the structure of ATP I have given because it is very important structure. ATP means sugar molecule with adenine base by N-glycosidic linkage. You know this thing. Now then 5 carbon, there are 3 phosphate. 1, 2, 3. First phosphate is linked with a low energy bond. This single line I have given, and there are 2 high energy bonds. So children, in ATP, you see adenosine triphosphate. How many bonds are there? 3 bonds are there attached to your 5 carbon. Clear or not? First bond is said to be your low energy bond and there are 2 high energy bonds. Now come to the one important question. Number of high energy bond in AMP, adenosine monophosphate, mean 1 phosphate. If I am talking about 1 phosphate, you see there is only low energy bond. High energy bond is 0. There is no high energy bond. Clear? So it will be 0. ADP, adenosine diphosphate. Children, you see 2 phosphate. So here 1 low energy bond and 1 high energy bond. So how many high energy bond? How many high energy bonds are present in ADP? The answer will be only 1. Clear? In ATP, there are 2 high energy bonds there. Very, very important questions. Clear? Now, another question. Which of the following is an example of dinucleotide? So that is answer will be FAD. Clear? So this is all about the biomolecules related to nucleic acid and important questions that can be asked in NEET and board examination. Now children, the differences between DNA and RNA. Have a look towards the board, my dear children. DNA is mainly double-stranded, whereas RNA is mainly single-stranded. DNA is double-stranded, but exception is found in 5x174 bacteriophage or bacterial virus, but the DNA is single-stranded. Very, very important. Similarly, RNA is mainly single-stranded, but the exception is found in reovirus, wound tumor virus, and a rice dwarf virus, where the RNA is double-stranded. But at least you remember two examples, that is 5x174 bacterial virus and reovirus. Okay? Now next point. DNA is fusion positive, whereas RNA is fusion negative. The most important point is DNA is fusion positive. Fusion is a stain, and DNA gives response to this stain or color, fusion, and gives you purple coloration to fusion stain. Clear? That's why DNA is fusion positive, but RNA does not show any response to this fusion stain. That's why it is called, RNA is called fusion negative. Very, very important question for NEET examination. Now next point, third number point you see, that is DNA is more stable, whereas RNA is less. Now, as because DNA is more stable, it provides more stability, therefore I have written here, it is DNA is preferred for storage of genetic information. Very, very important. Now RNA is your less stable, and it is better for, I have written, it is better for transmission of genetic information. Out of two nucleic acid, which one or which nucleic acid is better for transmission of genetic information? Answer will be RNA. And preferred as storage of genetic information if question asked, then it is DNA, my dear children. And these 2 points will be studying in RNA world, they are in NCERT. Absence of unusual bases, abnormal bases are absent in DNA, whereas in RNA there is presence of some unusual or abnormal bases. Examples of unusual bases in RNA are inosine, dihydrouridine, pseudoinosine, and 7-methyl-guanosine. Now, both DNA and RNA, my dear children, they show optical activity. Clear? They show optical activity. DNA is dextrorotatory, whereas RNA is left. But DNA having your high molecular weight and RNA has your low molecular weight. The rest of the differences I have already told while discussing the nucleic acid, DNA and RNA. Only some of the important differences that are being asked, that are normally asked in NEET and board,